Thank you very much, Daniel. Okay. Hello, everybody. My talk today will be about the development of methods and technologies to ensure safe and effective medicines. So in outline, I just want to touch on who safer medicines are, and I'll also discuss the current process for drug safety testing, why we need to innovate, and some barriers that we're facing to introduce these new technologies, and some progress that has already been made in this respect. And finally, I'll finish with the main asks from us today. So moving on, Safer Medicines Trust is a small UK independent patient safety charity. Our aim is to make medicine safer by encouraging a change in the way we test medicines based on animal tests to one based on human relevant science. And you can go to our website here for more information. So this is the traditional drug discovery process. Don't worry too much about the details, but essentially there's a research and preclinical phase before the line, and then a clinical in-human phase towards the after the line. And during that research and preclinical phase, virtually all those studies take place either in animals or are patient-free. And, touching on the point Neil and some other speakers have made, there's no real involvement in patients in that early stage. And that's perhaps quite astounding considering we're looking at treatments for human disease. But maybe not so much when we consider the output of those studies, with over 90% of drugs failing. And additionally, adverse drug reactions are one of the leading causes of death in the Western world today. And really, they're only truly manifested when medicines are released into the wider human population. And this means that we should really be thinking about retooling that whole drug discovery process and being more innovative with our testing methods to make drugs safer. Sorry about that. So, as I mentioned, many hundreds of drugs cause undesired side effects, and these can be pretty serious and lead to actually fatality. Over 10,000 deaths are reported to be due to ADRs in the UK every year, and these are not predicted by safety testing undertaken in animals or in early clinical trials. As a consequence, new drugs currently require prolonged testing in large numbers of people before they're licensed for use. And even then, warning labels may be issued. And there's a couple of examples here from the FDA for toxicity signals with drugs that are put onto the market. It's therefore imperative to develop better testing methods than we have now, and especially those that are relevant to human biology. While we may not have had the sort of technologies we have today available in the 60s, 70s, when primidos and sodium valproate were tested for toxicity, we do now have a wealth of platforms and methods which use human cells and tissues maintained under biologically relevant conditions of culture, which allow us to routinely study different human diseases and to explore disease susceptibility and to design and test novel drug treatments. These also allow us to predict and avoid human adverse drug reactions that cannot be detected in the animal studies, and alongside computational data analysis tools, enable us to predict accurately the human in vivo responses from these human relevant models. So in this slide, there are a number of publications describing human relevant models of various disease states. So you can see they look at various indications here. And there's also a recent review here looking at these so-called new approach methodologies, or NAMs as we call them, that can and are being used to study COVID disease. Okay, so the UK has been quite prolific of producing some documentation around and speaking to the development of new approach methodologies. But in reality, there's been limited implementation of these technologies in the preclinical regulatory testing space. The US FDA has produced a predictive toxicology roadmap in December 2017, and ICFAM, who are the Interagency Coordinating Committee on the Validation of Alternative Methods, has the remit to ensure that new and revised methods are validated to meet the needs of the US federal agencies. Likewise, the mandate for the European equivalent, ICFAM here, is specified in EU legislation on the protection of animals used for scientific purposes and includes a number of duties, including the coordination and undertaking of validation studies of alternative methods for safety assessment. So while we in the UK have the NC3Rs organisation and a non-animal technologies roadmap from 2015, this document is, in its own words, intended to guide the efforts of all those working in this area, not to actually enforce change. The vision for this roadmap has been described as that of a thriving sector that operates at the forefront of science, technology and innovation, and that delivers improved decision-making tools that result in more and rapid discovery and development of medicines, agrochemicals, chemicals and consumer products. However, I believe there are still a number of barriers to change before we can get to this vision. And in light of what we've been discussing today, I want to concentrate on the regulatory implementation of these technologies. So the EU, which we've talked about as well, which we're still part of until the end of the year, does still have a defined process to so-called qualify novel technologies for drug discovery, and for which applicants are encouraged to apply alongside the US FDA. There's a number of requirements to be met, after which, if successful, the EMA will amend the relevant guidelines and organise training. With the MHRA, we have the Innovation Office, through which we're asked to apply, but I don't believe there are any defined processes such detailing all the steps as yet. So finally, I'd just like to finish with a slide which shows a white paper the Safer Medicines Trust co-authored and launched in March this year as part of the Alliance for Human Relevant Science, which highlights what is needed from regulatory groups, the government, industry and funders to accelerate the growth of human relevant life sciences in the UK. You can download this from the link here, or I have copies which I can send if you wanted to email me. So in conclusion today, what we're asking for here is the ability to challenge any regulations which we believe are not fit for purpose, and to be able to propose new approach methodologies and solutions in a collaborative way with the MHRA. We request that clearly defined processes are made available to enable the adoption of innovative technologies into the medicine regulation process. The system must be agile and responsive in order to support innovation for safer medicines and ultimately enable the future-proofing of regulatory processes for safer medicines development. Thank you.